Hello, everyone, and welcome to today's webinar, Improving Heterologous Protein Production in Yeast with Massively Parallel CRISPR Genome Editing. I am Kaylee Bach of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Inscripta. To learn more, please visit inscripta.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well. If you have trouble seeing or hearing the presentation, please submit the questions below. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd now like to welcome our speaker, Eric Abbate, Director, Analytical Biochemistry, Applications Development at Inscripta. Eric, you may now begin your presentation.
Thank you very much for that introduction. Today I'm going to give you an overview of Inscripta's Onyx platform, and then I'm going to tell you about an application where we've used the platform in order to improve the expression of a heterologously expressed protein in yeast. So there are lots of challenges in the synthetic biology and biological engineering space. So if you look at it from, say, a forward engineering standpoint, where you're trying to produce more of a product or produce it faster, it really requires high-dimensional optimization. And unfortunately, we usually don't know the rules of biology, so we have to determine the improvements empirically. An example of this is production strain engineering. In the reverse direction, you have something like genome discovery, where you're trying to understand how the system works and trying to generate knowledge about that particular system. And in that case, you need to be able to take the system apart in order to re-engineer it for an intended purpose. An example of that is something like antibiotic resistance or gene profiling. So the scale and diversity pose a huge challenge to engineering biology. And the current methods that are used in order to introduce diversity into an organism are quite limited. So you might have things like gene knockout or gene knock-in type of experiments, or things like classical mutagenesis. But the challenges with these current methods is that they lack precision, they have slow and laborious workflows, there's a lack of diverse edit types, so a lot of these edits actually introduce loss of function mutations. And it's also difficult to separate hitchhiker mutations from real causal mutations for the phenotype that you're looking for. So on the axis here on the left, if you're looking at on the y-axis, say the variety of edit types from low to high, the number of genomic locations, also from low to high. In this quadrant where you have simple gene knockout or gene knock-in experiments, you're introducing a very low variety of edit types and you're targeting a few genomic locations. Something like protein engineering, where you're going to be introducing a very high number of edit types, but again, you're targeting a few number of genomic locations. Whereas something like genome-wide knockout libraries or classical mutagenesis, you're targeting a very large number of genomic locations, but the variety of edit types are quite low. So what you really want to do is to get up into this quadrant where you're doing true genome engineering and genome discovery, where you're introducing a wide variety of edit types at many different genomic locations. And that's where I'd like to introduce you to Inscripta's Onyx platform, or digital engineering, genome engineering platform. And it allows you to generate precisely engineered and bar-coded libraries of cells at your benchtop. So I'm going to go through some of the parts of the platform next. So you could do rapid genome scale engineering with the Onyx platform. And the platform really supports all aspects of the design, generate, test, learn cycle. And it all starts with the Inscripta Designer portal. And this is where a scientist can go in, and it's a simple graphical user interface, and design the edits they want to introduce in an organism anywhere in the genome. After you've designed your library and place your order, that initiates the manufacturing of the reagents and consumables that are necessary to build that library. In about 3 weeks' time, the reagents and consumables will arrive. You put them in the second part of the platform, which is the Onyx instrument itself. So you put those reagents and consumables into the instrument, along with the cells that you want to build this library in, and you press go. And in a matter of days, 2 to 4 days, depending on which organism you're editing, out will pop this tube of edited cells and this engineered cell library. And within this tube of edited cells, each cell will have at most one of the edits that you designed within your library within the designer software. And there could be thousands of different edits, and I'll talk a little bit about the specs later on. Inscripta provides kits that allow you to analyze that library that you built to see what the quality of it is. And then you can take that pool of edited cells and immediately go into some sort of test workflow. And this could be a screening type of workflow, or it could be a selection workflow as well. Once you've identified your hits from your screening workflow, or you've enriched in some population in a selection workflow, Inscripta also provide kits that allow you to extract the DNA from those samples and then submit them for DNA sequencing. That DNA sequencing information then gets imported into another software package called the Inscripta Resolver software. And this is what allows you to link the genotype of a hit to its particular phenotype. At this point, you'll probably come up with a bunch of other ideas that you want to test, or take a certain strain through another round that can be imported back into Inscripta Designer software, and you can go through this cycle once again. So the platform provides optimized designs. It utilizes the MAD7 proprietary nuclease. It has push-button simple workflow. You can design libraries anywhere from 100 to 10,000 designs per library. There's a single edit per cell. A wide diverse of edit types can be introduced, anywhere from 1 to 63 base pairs. And you could do rapid genotyping through barcode NGS. And an important point here is that it really democratizes genome editing. And I like to use myself as an example for this, because prior to coming to Inscripta, I had never designed a library. And I certainly had never built a library, but I had phenotyped a library. But by using the Onyx platform, I was able to come up with an idea, design a library based on that concept. Then once the reagents came in, I could put them on the instrument with the cells that I want to engineer and press go. And in a matter of days, out pops this tube of edited cells that I can immediately go into a screening workflow. So I was able to design, build, and then go into test of that library without help from anyone else, just interacting with the software and the instrument. There's a wide diversity of edit types that you can introduce into the genome. So this could target both the non-coding region and the coding region. So for the non-coding region, say in E. coli, you might think of doing a promoter and RBS insertion, maybe a 61 base pair insertion, and you could do this in front of every single gene in E. coli. For yeast, maybe you want to do a terminator substitution. So you could take a synthetic terminator and insert it or substitute it at the 3' end of every gene in yeast if you wanted to. And then as far as the coding region, one of the things we often like to do is generate knockout libraries. And we typically do this by inserting a triple stop codon at the beginning of the gene, or towards the beginning of the gene, in order to create a protein truncation. You can also do things like saturation mutagenesis, where you mutate every residue within a protein to all 19 other possible amino acids. And as far as some of the specifications, or more details about the specifications for the platform, so currently we support E. coli and S. cerevisiae, and this includes Inscripta standard strains as well as related customer strains. You can target the coding and the non-coding regions in the native genome, or if you've introduced a heterologous sequence, you can target that as well. And I'll talk about an example of that in the next section. As far as the designs per library, E. coli is anywhere from 1,000 to 10,000, and for S. cerevisiae, it's 100 to 6,000. As far as precision, currently there's one edit per cell that you can do. You can do insertions, you can do deletions, and you can do substitutions anywhere between 1 and 63 base pairs. The library composition that comes out of the instrument is typically greater than 90% of the designs are covered within that tube of edited cells. and anywhere between 15 to 75% of the cells are edited. The time it takes, reagent delivery is about 3 weeks, and library build takes anywhere from 2 to 4 days. But your hands-on time with the actual instrument is really less than an hour to start a run. Okay, so now that I've introduced the platform, I want to tell you a little bit about what you can do with it. And in this example, we're going to try to improve the production of a heterologous protein in yeast. So protein production is important for the bioeconomy. It has lots of applications in the industrial biotechnology space. So there are enzyme companies that produce enzymes for that can be used in the biofuels, in textiles, food production or processing, animal feed, household care like laundry detergents. There's also applications in the biopharma or therapeutic space where you might target, say, a metabolic disorder like diabetes using insulin, or in cancer therapies as well. And another really hot topic right now is the alternative protein market, where companies are trying to come up with non-animal sources of meat, dairy, or egg proteins. So the current tools to improve protein production are actually quite limited right now. And I used to work at an enzyme company where this is one of some of the tools that we used in order to improve the expression of a protein that we're interested in. So one common strategy is just to insert extra copies of the gene into the genome. And this definitely can improve expression, but the improvements often plateau once you insert many copies of that gene. You can start to play around with the expression cassette. So maybe you want to change the promoter. Very often you use a very strong promoter, but maybe there's a couple other ones that you might try. Or you can change the terminator. Or you can look at gene design, such as codon usage and codon optimization. So you might design a few different versions of it and see which one expresses the best. Or you could change the signal sequence if the protein is secreted. Another common strategy is classical mutagenesis of a production strain. You take your production strain, you treat it with a chemical mutagen, introducing a bunch of mutations throughout the genome. And certainly this can introduce beneficial mutations, but it can also simultaneously introduce deleterious mutations as well. And it can also be complicated to identify causal mutations for improved production because you're introducing so many different mutations all at once. And what we think is that the Onyx platform provides an attractive alternative way of improving protein production. So in order to come up with a way to test this, we had to come up with a model system. And what we decided to do was to try to improve the expression of an enzyme called CBH1. So CBH1 stands for cellobiohydrolase 1. This is an enzyme that's involved in the degradation of cellulose, and it releases cellobiose from the reducing end of a cellulose chain. Essentially, it works as an exocellulase. So the CBH1 works in conjunction with a lot of other enzymes in the efficient degradation of crystalline cellulose. So you can see the CBH1 here working on the reducing end, cleaving off cellobiose subunits, which is just two glucose molecules linked together. Then an enzyme called beta-glucosidase takes those cellobiose and converts it ultimately to glucose. And why do we care about CBH1? Well, it turns out it's important for an idea called consolidated bioprocessing, or CBP. And in a CBP process, the idea is that you have a single strain that produces the enzymes that are necessary for the degradation of some sort of input feedstock. Very often it's some sort of lignocellulosic waste plant material. The enzymes degrade that feedstock, converting it ultimately to a sugar, which is then utilized by that same strain in order to produce some sort of fuel or chemical. So essentially all of these enzyme production, substrate hydrolysis, and fermentation are all consolidated in one single reaction. So that's why we wanted to, one reason why we wanted to try to improve expression of the CBH1 enzyme. So the first thing we had to do was, to generate the CBH1 expression strain and develop an activity assay. So we decided to use a fungal CBH1, and we did codon optimization of that gene for expression in yeast. We used the native fungal signal sequence for that CBH1. The expression of the CBH1 is driven by a yeast ENO2 promoter, and we utilized a DIT1 terminator. As far as the base strain we used, it was based on what utilizes SenPK yeast strain, and we generated a single copy genomic integration of the CBH1 expression cassette into that strain. The assay is actually pretty simple and straightforward. So it utilizes a synthetic substrate called PNP beta-lactopyranoside, and which is shown over here. So the CBH1 actually cleaves this bond right here, releasing paranitrophenol. And at elevated pH, paranitrophenol has a characteristic of absorbance maximum at 405 nanometers. So we can just measure CBH1 activity at 405. And we utilized this activity assay as a proxy for active enzyme production. I should mention that there's no detectable activity in the parent SenPK based strain. Okay, so the screening workflow that we used to test the various libraries that we're going to build on this production strain is pretty simple and straightforward. So we took that tube of edited cells that come out of the instrument, we plated it on agar plates, and then we would pick colonies into 96-well mid-well plates. And in a typical screening workflow, I would pick between 10 to 20 plates per library. And these were picked into 300 microliters of YPD, so a rich medium was used for expression. These plates would be grown to saturation for approximately 36 hours at 30 C. Then we would subculture those plates into the production plates, which is essentially just fresh YPD media. And then this production would go for 24 hours. So the saturated cultures were then stored as glycerol stocks at -80. So after 24 hours, we would measure the CBH1 activity in the supernatants for our primary screen. Once we did that, we would identify hits. Those identified hits can then go into a retesting of them to confirm that they're indeed hits. We'd go back to our glycerol stocks that we have and go into a retesting protocol, which is essentially going through the same process again, but this time they were tested in quadruplicate. Once we confirmed the hits, we would then genotype. So just to show you a little about the noise within the screening workflow, we would pick, I picked 96 colonies of the base expression strain, and then I would take it through this entire screening workflow. And then the results of that experiment is shown over here. So the average absorbance at 405 nanometers for those 96 samples, 0.26, with a very low standard deviation, and a CV of 1.7%. So the noise in this screening workflow is extremely small. So this really allowed us to pick out hits with improved CBH1 expression with very small improvements in CBH1 activity. Okay, so now we needed to come out with what type of libraries and what concepts do we want to test to try to improve CBH1 expression. And it kind of broke down into two different categories. So we had genome-wide libraries. So this is basically targeting everywhere in the genome. And then we had targeted libraries that were targeting either the CBH1 expression cassette or targeting certain genes that we thought might be important for CBH1 expression. So for the genome-wide libraries, we had genome-wide knockout libraries where we insert a triple stop towards the beginning of a gene, and we did a knockout library of every single gene in yeast. And then we also made genome-wide synthetic terminator insertion libraries. So we took a synthetic terminator, inserted at the 3' end of every gene in yeast. And then we also had genome-wide deletion libraries. So in this particular library, there were deletions between 1 and 59 base pairs that were introduced throughout the yeast genome. And these deletions could target either promoter regions, or it might target within the open reading frame of a protein as well. Then we had on the targeted library side, we had a couple different variations that we designed. And several of them, or a few of them, targeted the ENO2 promoter itself by targeting the predicted transcription factor binding sites within the ENO2 promoter. And I'm going to talk about some details of these libraries later on. We also made a CBH1 alternate codon library that I'm calling, which is essentially a site saturation synonymous mutation library. Essentially what we were doing was we were taking every codon in CBH1 and changing it to all alternate codons that encode for the same amino acid. Essentially, we were trying to do codon optimization of the gene. or see if we could do further codon optimization of the gene. Then I have another library that I'm calling the targeted knockout library. And for this library, I mine the literature to try to find genes that have been previously identified that when they're knocked out, they can improve the expression of some heterologous protein. So I threw all those into one library. I also did a genome-wide protease deletion library where I knocked out every protease, or targeted the knockout of every protease in yeast. And then also targeted the knockout of some enzymes in the glycosylation pathway, which could have an impact on the secretion of the CBH1. Okay, so these are the different library concepts that we wanted to test. So what I'm showing here is the primary screening of these libraries on CBH1 expression. And this is showing it as a box plot. And on the y-axis, I have the fold change over the base strain. And these are all the different concepts that I just described, broken down into both the genome-wide type of libraries as well as the targeted. And in all of these cases, all these different libraries that we produced, you can see that there are many hits that we identified that had a significantly improved expression of the CBH1 enzyme. So these hits were then taken into a retest workflow that I described, where they're tested in quadruplicate. And that's shown here. And we identified hits ranging from a 1.1x to a 1.6-fold increase in CBH1 activity. I should mention this data that's shown here in the bar graph. I'm showing the fold change over the base strain. And the bars are showing the average and the standard deviation of quadruplicates if we identified a hit only once. If we identified the same hit multiple times in a library, then I averaged the fold improvement of those hits, and I'm showing the standard deviation of that. I somewhat arbitrarily picked a hit as being anything greater than or equal to a 1.1x over the base strain, which is something that we could reasonably pull out based on the very low noise within our screening workflow. And you can see that the two strategies, whether it's genome-wide or targeted, we were able to identify a bunch of different hits from them. Some of them into the 1.65, 1.5-fold for nearly all of them. And it was nice to see that even with the CBH1 alternate codon library, we were able to pull out some really nice hits from that. Okay, so one of the things I wanted to talk about at this point is phenotyping strategies and what really drives your phenotyping strategy as far as how many plates should I sample of these different libraries. And one of the concepts that you need to talk about is phenotyping library coverage. And this is really the, the amount of your library that you're able to screen when you take through a certain number of isolates through the workflow. So the phenotyping library coverage is really dependent on several different factors. And it's the number of total designs within your library. It's the edit fraction of your library. So not all of the cells that come out of the instrument are going to contain an edit. Excuse me. So that editing fraction really is important to determine your coverage. The distribution of the edits within your library. So this is also important because not all edits are going to be equally represented in your library, and certain edits might be overrepresented than other ones. And then the other important factor is the number of samples that are screened. And what these factors allow you to determine is what we're calling a screener score. So an example of that is shown over here on the right, and this is shown in the Resolver software. In this particular example library, it's about a 5,000 member library, and the editing fraction was about 69%. And what's shown here on the x-axis is the number of isolates that are screened and the percent of your library that you're actually covering during that screening workflow. So let's say we were going to screen about 5,100 isolates. With 69% editing, you would end up screening about 31% of the total number of input edits within your library. So 31% of the 5,000 members would be screened if you screen 5,100 isolates. It's important to note here that the slope of this curve isn't constant. So at low sampling, the slope is quite steep, whereas it's much more shallow when you get to higher levels of sampling. And what that means is that as you get to really high levels, the more you go up in the number of isolates you screen, the marginal improvement gets smaller and smaller as far as the number of the library that you're covering. So in this region, you're getting a lot of bang for your buck. You're getting much less bang for your buck once you start get up into this region. So this concept went into generating this table here. So what I'm showing is the library concepts that were being screened, and then the library size of each one of those. So in total, there were about 15,000 edits that were designed in an attempt to improve the expression of the CBH1 between these 7 different libraries. And then I'm also showing the number of isolates from each one of these libraries that is screened. In total, there was about 8,500 isolates screened. I'm also showing the estimated library coverage that was screened for these different libraries. So you can see some of them have fairly shallow sampling. So the large libraries, you're screening only about 3% to 13% of the total diversity. Whereas some of the smaller ones, because they were screened at a higher level, you're getting closer to the maximum amount of the library that's being screened. I'm also showing the number of retest hits from each one of these libraries, and then also the number of unique hits. So in some instances, we pulled out the same hits multiple times from a library. And then I'm also showing the hit rate here. And the hit rate can kind of give you an idea of how fruitful that particular library is in identifying hits. So with a low hit rate of about 0.5%, the CBH1 alternate codon library, even though that's a somewhat low hit rate, we're still able to identify hits from that. And one of the important points that I wanted to bring up here is that shallow screening of large libraries can identify strains with improved phenotype. So you can see that even though I screened the genome-wide knockout and the terminator at only 13 or 3%, I was still able to pull out a large number of hits and a large number of unique hits, providing a lot of diversity that can be used for further library builds. And I think an important point that I want to make as well is that with limited phenotyping capacity, it's better to shallow screen several libraries than screen a few very deeply. And the idea behind this concept is that each one of these libraries is testing out a different strategy or a different way of improving CBH1 expression. And it's far better to screen a lot of different libraries at a shallow depth with limited phenotype capacity than to, say, screen a few of these concepts at very deep depth, because screening more libraries allow you to explore lots of different concepts and lots of different diversity for improving the expression of your heterologous protein. Okay, so if I wanted to look at a lot of these hits that we identified in these libraries that target kind of the genome, or basically the expression strain itself, it was interesting to see that they fell into different categories involved in different cellular processes. So I have it broken down in a stacked bar chart here by the different libraries: genome-wide knockout, deletions across genome, the genome-wide terminator, and the targeted KO library. So we saw a lot of hits targeting proteins involved in the protein degradation pathway. Several of them involved in protein trafficking, so it's likely important for secretion. Lots of them involved in genes involved in stress response, involved in transcription or translation, nuclear transport, glycosylation pathway. A few of them fell into this basically an other category. Some of them were involved in metabolism. And then we also identified several hits that was interesting, that they were gene of unknown function. So this was kind of interesting that we were able to pull out hits in this category, because through rational strategies of targeting certain genes, you would have never targeted a gene of unknown function for improving protein production. So in doing an unbiased sort of genome-wide type of strategy allows you to pull out hits that you would have never identified if you had used a rational strategy. So looking at some of these hits that we pulled out from these different libraries, it was interesting to see that in some instances we were able to pull out the same hit, or an edit targeting the same gene, in different library strategies. So for instance, this UBP8 gene, which was pulled out of the deletions across genome library, as well as the targeted knockout library. And it's important to note that these are actually two different strategies. So the targeted knockout introduces a triple stop at the beginning of the gene, so the gene basically approaching truncation. Whereas in the case of UBP8, there was about a 50 base pair deletion in the middle of the open reading frame. And both of these libraries were able to yield this UBP8 gene, and it was very nice to see that the fold improvement from this gene, or edits targeting this gene, were very similar. The same was true for STM1, and then also for SUM1 as well from the genome-wide knockout library, as well as deletions across the genome. And it was very nice to see that the fold improvement from these were very, very similar. So this really gave us, nice confidence in our screening workflow that we're pulling out hits reproducibly from multiple different libraries, and also the noise level in our screening workflow, because these libraries were built at different times. The screening of these libraries was done on different days at different times, and yet we were able to pull out the same hit with a very similar fold improvement. Okay, now I wanted to talk about the libraries that target the ENO2 promoter itself, so the promoter that's used to drive the CBH1 expression. And we wanted to see if we could use Onyx libraries to investigate the promoter architecture. So I'm showing the CBH1 open reading frame here, as well as about 500 nucleotides of the ENO2 promoter upstream of the CBH1 gene. And what we wanted to do in these libraries was to target the predicted transcription factor binding sites located within the ENO2 promoter. And we target these binding sites with designs testing out a couple different strategies. And that's what's shown here on the bottom of this graph, are the different design strategies. So the first design strategy was an insertion strategy, where we take the native transcription factor binding sites and we add extra copies of them adjacent to where the native ones are located. And that you can see the locations of them right here, targeting all these different regions. Then the next strategy was to delete those binding sites individually. And you can see those are the different deletions here in blue. Then we also made substitutions or swaps. And in that case, we took a pool of 109 transcription factor binding sites and swapped them in at the native binding sites within the ENO2 promoter. So we're covering basically this whole region with edits. So on top of this black line is shown the hits, or the location of the hits that were identified. I'm going to zoom in on the next slide in this region so you can see it more closely. But what I wanted to point out was it was interesting that we targeted lots of binding sites within this region, but we didn't pull out any hits at all that were in close proximity to the start of the CBH1 gene. So if we zoom in on the location where we identified hits, again, we're showing the designs down here in insertions, deletions, and substitutions by the different colors. And then the hits are identified above the line. So there are several different binding sites located within this region of the ENO2 promoter. So there's a few ASH1 binding sites, MOT3, FO2, HSF1, GCR1, and GAL4 binding sites. If we looked at the hits that we identified, so kind of walk you through some of these. So at the MOT3 region, if we swap in an HAA1 transcription factor binding site, we were able to see improvement of CBH1. I should mention, if there's multiple boxes at a location, that means that we pulled out different types of hits at that location. So this ASH1 binding site is kind of interesting in that we actually got hits from all three different libraries at this particular location. So if you delete the ASH1 binding site, you improve CBH1 expression. If you substitute in several different binding sites, you improve CBH1 expression. Or if you insert a MOT3 binding site right next to the ASH1 binding site, you also see improvement of CBH1 expression. And what this suggested to us is that this binding site might actually function as a repressive site for CBH1 transcription. In that if you delete the binding site, swap in another binding site, or put a different binding site in very close proximity, you're able to improve the expression of CBH1. At FO2, we found a lot of different swaps improved expression. An insertion of FO2, an extra FO2 site here, some swaps here at the ASH1 binding site, or insertion of extra GCR1 sites at this location could improve expression as well. So we basically got hits from all three of these different concepts. And I think what's a nice point to mention here is that this is all happening in the context of the genome itself. So we're doing, promoter studies in the context of the actual genome. It's not on a plasmid, which might not be incorporated into chromatin as normally would happen in the genome. So this might give you a more accurate representative of the changes to expression at the promoter. Okay, so I kind of went through some of the small scale screening workflow. Now we wanted to see, well, we identified all these hits. Well, did any of them scale up to a larger culture? And we tested them in 25 ml shake flasks using YPD, just as we did in small scale, and 20 grams per liter glucose, exactly the same conditions that were used. So we took a series of hits from our libraries, so things coming from the genome-wide knockout, to insertions or swaps, and then a CBH1 alternate codon library, and we grew them up in shake flasks. And I'm showing here the growth curves from these experiments. And because we had larger cultures, we could do time courses. And I took samples at 9 hours, 24 hours, 48 hours, and 72 hours. After 48 hours, when I thought the glucose was probably fully consumed, I added an additional 4 grams per liter glucose to see if I could improve the expression of CBH1 further. So this is showing the absorbance at 600 nanometers versus time, basically looking at the different growth curves. And the base strain here, which is shown as a black dotted line, you can see it right here. It basically has a normal growth curve. Several of the hits have very similar growth curves, but it was also interesting to note that many of the hits had lower growth curves, although not significantly different. But you can see that it definitely grows less well, or grows to lower OD than the base strain. So a couple important points here is the reason we saw improved CBH1 expression or CBH1 activity can't be explained simply by you just have more cells. If these edits cause the strains to grow better, you have more cells producing more enzyme. That can't be the explanation. And it was also interesting to note that several of the improved strains had lower growth curves than the parent strain or the base strain. And there's some evidence in the literature that people have seen this in hits that they identified for improved heterologous protein expression. And the thought is that some of the carbon that's usually going towards more biomass production is being diverted to improve protein production. So it's interesting that we saw that in several of the hits here. Now, if we look at the actual CBH1 activity for this time course, again, the base strain is shown in here as a dotted black line, and then all of the other strains here. And it was very nice to see that all of the hits that we tested at large scale are also hits at large scale. All the hits that we identified in small scale also ended up being large hits, hits in large scale. And you can actually look at some of the growth profiles or the production profiles and see how it compares to the base strain. So some of them, like for instance these two right here, they actually have very similar profiles. They just grow up or they produce slightly higher levels of CBH1. Some of the other ones, like for instance these two right here, they actually produce at higher levels at the early time point, but then they level off kind of at a middle range for CBH1 expression. And then, this strain right here, which kind of produces in middle range at the beginning, it just levels off at a much higher level of expression. And the addition of extra glucose impacted some of these for more CBH1 activity, where in other instances it had no impact. But it was very nice to see that all of these hits scaled up, which gives us, again, greater confidence in our high-throughput small-scale phenotyping. I also wanted to look at what is the fold improvement that we get in flasks versus plates. And I picked this 24-hour time point in order to do this calculation because those are the time points that were in common between the two scales. And what you could see from this data, looking at the percent improvement in CBH1 activity, CBH1 expression, is that in some instances, there's a very similar fold improvement. In some instances, you actually see a much greater improvement in CBH1 activity. So the largest hit here was an 84% improvement, going from a 57% in small scale. And then some instances, we saw a smaller fold improvement. But overall, it was very nice to see that all the fold improvements or the improvements translate in larger scale. Okay, so now if we look at the type of libraries and the cellular processes that were targeted by these libraries, or the hits that we identified, there were some interesting things to know. So we have these genome-wide knockout and deletion libraries targeting in the genome. We had genome-wide terminator libraries. We had libraries targeting the ENO2 promoter driving expression of CBH1. We had alternate codon libraries of CBH1, protease knockouts, and glycosylation knockouts. The hits actually target lots of different cellular processes. So the hits targeted transcription, whether it's sort of global transcription or transcription from ENO2, nuclear export, stress response. So you might imagine protein expression can cause a lot of stress for a cell. Translation, protein degradation. So we saw lots of hits that regulate proteasome function or just individual protease knockouts, or secretion, including glycosylation knockouts. And I think an interesting point to note here is that all of these processes that we're targeting, or edits that are targeting these different processes, all of these processes are likely important, or what you might expect are important for the expression of a heterologous protein. And one of the exciting things I think from this is that each one of these pathways, it likely has a bottleneck for CBH1 expression. And it gets me excited about what happens when we start combining these different edits into a single strain, where you're now alleviating multiple bottlenecks for a protein expression. So hopefully the combining of these edits into a single strain will be the topic for a future talk. Okay, so the last point I want to talk about is the time invested in building these libraries and going through this type of project. And that to try to prove to you that the Onyx platform really enables rapid learning cycles. So the first thing we had to do was generate and QC the CBH1 expression strain. Then we used the designer software to design the libraries, and then the reagents were manufactured. Then you have to build the libraries, and then you have to phenotype and genotype those hits, or the libraries, and then subsequently the hits. So that's going to be shown in the bottom of this slide by these various different colored arrows. So I'm showing sort of a sample of a project timeline for this project. And this is broken down into 1 month increments. So the first part of the project was to build this CBH1 expression strain, which after we got we figured out which promoter we wanted to use and terminator and all the details of how to express the CBH1, it took about 1.5 to 2 months in order to build this. Now, obviously, this timeline can be highly variable. It could take longer than that, or if someone already has their production strain or their expression strain, then there's no time in order to produce that. Then to design and manufacture the 7 different libraries that were screened, it took about 3 weeks in order to do that. Once those reagents arrived, you have to start building those libraries. And if you have one instrument, you have to build those libraries serially. So in the case of this particular strain of yeast, builds take anywhere from 3 to 4 days. So I could build about 2 libraries per week. Once you build your libraries, then you can go into phenotyping. And the strategy that I used was that I would phenotype 2 libraries, or initiate the phenotyping of 2 libraries per week. So after the first week, I would have 2 libraries built, and those 2 libraries would then go into the phenotyping workflow. And the entire phenotyping and genotyping of the hits took about 1 month for each one of the libraries. But they could be done in parallel. So I would initiate the phenotyping of these 2 libraries, And then during that same week, I could start building the next set of libraries, which could then go into phenotyping the following week. So again, you could build a library serially, but the phenotyping and genotyping could happen in parallel. And ultimately, it took about 3 months in order to design, build, and then phenotype and genotype these libraries with this particular strategy in mind. And in total, the 7 libraries were screened and 74 unique hits were identified for improvement of CBH1 expression. I also want to bring your attention to the resources that were involved in order to do this project. So designing a library can take anywhere from half an hour to a few hours per library. The build, the amount of time you have to actually interact with the instrument, putting the reagents and your cells on, it's really only a few hours. So maybe 2 to 3 hours per week is spent on that. And then as far as phenotyping and genotyping, I estimate it's about a quarter of an employee to do this particular workflow. So not a huge amount, it's actually a fairly small amount of resources in order to identify all of these hits in this small period of time. Okay, so as far as conclusions, kind of take-home messages are the Onyx platform is really a game changer for DGTL cycles. So it allows you to rapidly design and build genome-edited libraries using significantly less resources. And it really democratizes genome editing. Again, I use myself as an example for the CBH1 project. I was able to come up with a concept, design that library, build that library, and phenotype it all on my own without anyone else's help. I also want to make a point that genome-wide or untargeted libraries and targeted or rational libraries can both identify strains with improved protein production. So we found both of those strategies as being very fruitful, targeting the expression set or doing rational knockouts, as well as untargeted and unbiased genome-wide libraries. And also that shallow screening of large libraries can yield beneficial diversity in that phenotyping a wide variety of library concepts is likely better than deeply screening a few libraries, particularly when you, in the next step, when you want to start combining those, since they're targeting potentially different bottlenecks within the protein production pathway. Once you start combining those, you might be alleviating multiple bottlenecks and be able to obtain strains with significantly improved expression. And with that, I'd like to thank you for your time, and I'd be happy to take any questions that you might have.
Thank you, Eric, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Okay, let's get started. Our first question asks, can you upload in Inscript Designer your genome of interest? Would the edit protocol work with organisms or cells other than Escherichia and Saccharomyces? Excuse me for the pronunciation.
Yeah, good question. So indeed, you can upload whatever genome that you're interested in or your genome into the designer software. The key there, though, so you can upload it and then you can design edits against that. The key is that currently we're only supporting E. coli and Escherichia for that process. So you can't just use whatever cells you want to use. It has to be a strain that's similar to one of Inscripta's standard strain. And there are kits that we provide that allow you to actually test the editing process in your particular strain. But currently, E. coli and Cerevisiae are the two hosts that we're currently supporting.
Great, thank you. Our next question asks, genotyping is by sequencing amplicons?
Yes, so there's a couple different strategies you can use. You can either sequence the barcode to identify the edits that are contained within that hit, or you could do whole genome sequencing. So we've used both strategies in identifying hits. So both of those are possible.
Great, thank you. All right, next question here. Your normal application here is CBP. which looks nothing like MTP or shake flask growth? Which library designs are likely to yield hits that are productive in application? More broadly, how important is it to screen in application relevant conditions?
Yeah, for sure. I mean, that's a very important point. And this is kind of a scaled down version of a CBP, type of process, in that we're really trying to improve the expression of one enzyme that would be involved in CBP. This is something that if you're actually looking for, a real CBP type hit, you would want to be doing this in a more complex system where you're expressing many other enzymes. This is kind of a simplified case that allows us to use it as a test case. Certainly, you would want to screen as close to application conditions as you possibly can get. I mean, this is something that is very common, that microtiter plate or small scale experiments. You need to make sure that those hits go to more application relevant type processes. So that's something that you have to do. And in this case, we're not actually trying to build a CBP strain. We're just trying to show this as an example of a way to improve the expression of an enzyme. But that's definitely an important point, is that you want to screen in conditions that are most relevant for your application or for scale up.
Wonderful, thank you. We've got lots of great questions coming in here. The next one asks, is deep screening ever warranted? Does the hit analysis tell when it's worth sampling deeper?
Yeah, so that's kind of an interesting question. And the shallow screening strategy, I think kind of really depends on what you're trying to get out of your library, right? So there might be an example where you have, say, a really targeted library. So we have this targeted knockout library that we wanted to study. And maybe in that case, you're really trying to answer the question of, do these hits have any impact? And in that case, you might actually want to screen quite deeply to make sure that you've assessed all of those hits and whether or not they impact the phenotype that you're looking for. And then it also depends probably on the amount of hits that you get out. So if you're able to shallow screen, say, a large library and you get a large number of hits, then maybe you're happy with that number of hits. Because kind of one of the keys that you have to think about is you want to identify hits as fast as you can, which usually yields some level of improvement. But the large level of improvement really comes when you start combining those hits into one strain. And this has been seen in protein engineering type of experiments, as well as in strain engineering. We have a nice example of this with lysine production, where we saw the really large improvements when we started to combine those hits. So I think it comes down to something where what is the phenotyping capacity you have that's available, how many hits you're identifying, and how fast you want to move. Because maybe you just want to identify some number of hits and then just really start combining those. So it's a good question, and it really depends on what strategy you're trying to address, or your strategy, your phenotyping capacity, the speed that you want to work. There's lots of factors that kind of go into that decision. Good question.
Great. All right, next question here. How does the pipeline change for an adaptively evolved E. coli or S. cerevisiae strain?
Yes. So I'm not exactly sure what they're trying to ask. I assume they mean if you have a strain that you've done ale type of work on, how does the process change? And if that's the question that's being asked, so usually you have to do some sort of genome sequencing of that strain so that you can upload that into the Inscripta designer platform, or the Inscripta engineering platform, so that you can more accurately design the edits against that strain. So ideally, you want to have good sequencing information. I think that's really critical.
All right, our next question here says, you haven't yet had a chance to build the combinatorial libraries. Do you have an example of how high an expression target could be improvised based on the addition of positives from several areas of physiology? What would be a reasonable guess as to how high the activity could go as a result of combinatorial effort?
Yeah, so that's a difficult thing to assess right now in this situation. I mean, we do have examples in this previous application I mentioned where we were trying to overproduce lysine in E. coli. And we got a lot of different hits throughout the genome, whether it's targeting the lysine biosynthetic pathway or just genome-wide things that are targeting basically the genome everywhere. And what we found in that case was that we didn't start seeing really large amounts of improvement until we started combining them. And the greatest improvement we saw was about a 14,000-fold improvement when we had 3 different edits that were combined within that strain. Now that's for a small molecule. So I think that's likely not what you're going to see with the protein production. You're not going to see a 14,000-fold improvement. It's unlikely. But I think it's difficult at this point to really say because we're starting with probably a very unoptimized system. We're using a wild type yeast strain that we've inserted a gene, so not heavily optimized. So my prediction is that when you start combining those, that you're going to get a much larger level of improvement, but it's hard to say at this point.
Great, thank you. Looks like we have time for a few more questions here. This next one asks, what is the advantage of using the InScripta platform for genome-wide knockouts over, for example, transposon mutagenesis?
Yeah, so for transposon mutagenesis, I believe those can be targeted. I'm not sure that they can actually be specifically targeted. They might be able to, but I think one of the advantages of, I think, the InScripta platform is that you can specifically target it to a location. And it doesn't even have to be to a specific gene. You can target it anywhere within that gene because you have the opportunity to target so many places with the gene. And using CRISPR, you can really engineer the location of where that cut's going to be introduced. So I think one of the advantages of this platform is it's really targeted. And then it's also a trackable system. So you can have a barcode that allows you to very rapidly genotype and track those edits that are being introduced.
Great, thank you. And we have time for one more question here. Can Onyx generate random multiple genome edits in one cell simultaneously?
Yeah, so random genome edits can't really be introduced. I mean, it's all targeted, right? So I guess you would say that's not necessarily random. And currently the platform allows for the introduction of a single edit at a time. But I will say that we're working on expanding the platform to be able to introduce multiple edits at the same time. And in that case, you can do maybe combinatorial type of libraries where you can have different registers that allow you to introduce a combination of different edits simultaneously, which allows you to build larger combinatorial type space or allows you to sample larger combinatorial space, much greater than a single edit would be able to introduce.
All right, well, thank you again, Eric, for your time today and your important research. We would also like to thank LabRoots and our sponsor, InScripta, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.